Okay, today we're talking about photosynthesis. Okay, so first of all, does anybody have a good summary, a good definition of what they think photosynthesis is or how you would define it? So, what I would say for this definition, and you can keep typing, what I would say for this definition is that photosynthesis is the process by which sunlight is used to convert carbon dioxide and water into glucose and oxygen. So this is how I would define our reaction here, photosynthesis. So we are using the reactants, carbon dioxide and water. We're getting energy from sunlight and producing the products, glucose and oxygen. So in this way, the plant will take the electromagnetic energy, the light energy, and convert it into chemical energy in the form of the chemical bonds in glucose. Yes, that is exactly right. The photosynthesis light cycle produces glucose, and then that gets used for cellular respiration. So they go back and forth. So let's think a little bit about why plants might need photosynthesis. What sort of factors play into why a plant needs to convert sunlight into glucose? They don't eat sugar. That is absolutely correct. They also can't typically move. They are usually rooted in one spot unless they are have very specific adaptations. They are typically autotrophic, meaning they produce their own energy. They don't consume energy like we do. We get all of our energy from consuming things. Plants produce their own energy. And then, and this is with the exception of carnivorous plants. So plants that do actually consume other organisms for food. But they typically do that for nutrients as opposed to energy itself. So let's go ahead and put the chemical reaction up, and we'll talk about that a little bit. They do need CO2 in the environment to survive, and we're going to talk a little bit about how they get that. First, let's talk about the equation itself. So you can see here, this is a just a written out version of the equation. This and the definition that I gave you say pretty much the same thing. So on our reactant side here on the left, we have our carbon dioxide and water. Then we have sunlight, which is our energy provider, is our energy source. And then we have our product side, glucose and oxygen. In some cases, you're going to see water written on both the reactant side and the product side. We're going to talk about that in just a second. So let's talk about these little quirks about this reaction. So first, why is sunlight not considered a product or a reactant? Any thoughts on that? Because it's definitely an essential part of the reaction. So yeah, so it's partially because of the nature of light itself. Yes, and it's used to provide energy for the electron transport chain. So the reason it's not typically considered a reactant or a product is because we're not changing the light. The light is being used, it's being captured, but it's not being broken from one shape into another shape in the way that the carbon dioxide, water, and glucose are. So it's not being changed, it's just being used. So that's why we write it above the arrow in much the same way that we write enzymes and catalysts above. Now, why would water be on both sides? Why could we have water on both sides of the reaction sometimes and on one side other times? Yes, so the reaction is using the hydrogen and the oxygen from the water, but it's taking these molecules and it's rebonding them in other arrangements, and we end up with water as a product as well. So in your McGraw-Hill equation, you're going to see this same equation, but you're going to see it with, hold on just a second. You're going to see it with water on both sides. And on the reactant side, you're going to see a 12. And on the product side, you're going to see a 6. So what the equation that you see in McGraw-Hill is saying is that we're using 12 molecules of water and we're producing 6 molecules of water. So there's more water being used in the reaction than being produced. So why would we end up seeing some reactions where we only have the water on the reactant side? So it's a net reactant. So we're pretty much canceling out the molecules that have been used and produced when they balance each other out. So we go from having 12 on one side and 6 on the other side to having 6 on one side and none on the other side. So we're just not showing the water molecules that balance each other out in the reaction. This is called a net reactant, or the net amount of water. So it's there. It's definitely both a reactant and a product, but depending on who's writing the equation, who's talking about it, and what reasons we're talking about it, you might see it written in different ways. Does that make sense? Okay, so now we know the chemical parts of this reaction. We know the reactants, we know the products, we know the role that sunlight plays. But how does this actually work practically in a plant? What is the actual mechanism that we're looking at here? So as we know, there are a few set parts of a plant that we almost always pay attention to. So we have our roots, we have our stems, and then we've got our leaves, right? So these are the most commonly talked about parts of plants. Okay, so anywhere in our plant where we have green pigmentation, we're going to be seeing photosynthesis occur. And the reason is because of the organelle that undergoes photosynthesis. Does anybody remember which organelle that is? Yeah, there we go. Yeah, you guys are right. It's the chloroplasts. So chloroplasts are the organelle responsible for photosynthesis. And they are most often found in like stems and leaves. And in trees, they're not going to be found in the stems because they are woody stemmed plants, but you'll see them in stems of like herbaceous plants, like grasses or flowering plants that you might see in a garden. So any of the green tissue is going to contain chloroplasts, and those are going to be photosynthetically active. So what part of the chloroplast is actually doing the work, though? So chloroplasts contain proteins, and these proteins are called photosystems, and photosystems contain chlorophyll. So they contain proteins that hold chlorophyll. Now, chlorophyll is a pigment. And it's not the only pigment that's found in these chloroplasts in the photosynthetic pathway. So there are actually 4 different pigments that are most often involved. So there are 2 types of chlorophyll. There's chlorophyll A and B. And then there is carotene, or carotenoids. You might hear it called a carotenoid. And then there is xanthophyll. So chlorophylls are going to be the green pigments. And then anybody have a guess? That's exactly right. The green pigment chlorophyll cannot absorb green light, and we will get more into that in just a few minutes. Does anybody have any idea what color these other 2 pigments might be, carotene and xanthophyll? We are actually going to be seeing a lot of these right now during this particular time of the year. Maybe not so much here, but in some places. Yeah, so we've got like reds and oranges. These are really common in fall because the plants are going to be pulling back energy to go dormant for the winter. So the chlorophyll gets reabsorbed first, and then you see the orange pigments of the leaves. So carotene is going to be the orange pigment, and xanthophyll is going to be the yellow pigment. It's going to be more like orange-red. So these are our pigments. We've got chlorophyll A and B, carotene, and xanthophyll. These are all going to be involved in absorbing the light. So these pigments act sort of like antennas. They're going to be picking up those light waves and then converting that into the energy that's used in the reaction. And the way they can do this is by electrons. So somebody in the chat already mentioned how the light stimulates the electron transport chain. That's exactly right. So these pigments have electrons that when they get hit with light, the light brings the energy level of the electron up, right? So it gets to a higher energy level. And then once it's at that high energy level, it's going to bounce back down to the lower energy level. And as it goes back down to the low energy level, it's going to be giving that energy to the rest of the reaction. So the pigments are the antennas. The electrons are going to allow that sunlight to be picked up and transferred to the actual reaction. And yeah, so that's going to be sort of the like practical application of this. So now we know about the pigments, which ones are going to be responsible. We know where the photosynthesis is happening in the plant. So let's talk about how the plant is going to get all of its reactants. So we know that when we need energy, we can go grab a snack, eat a meal, drink some water when we need water. So how do these plants who cannot move, have no conscious thought, how are they getting all of their photosynthetic reactants? So first, let's talk about water. So where is the water coming from in a plant? So they get their water through the roots, and it's actually pulled up through the plant. This is from a process called transpiration. Pulled up from the roots, from the soil around the roots. Transpiration. Right? So it's coming up from the soil. So that can come from you watering your plant, that can come from the water table in the natural soil, that can come from rainwater, anything like that. Let's talk about carbon dioxide. So we had this other question earlier. The plants need carbon dioxide for this reaction. Where do they actually get it from? So where do we see carbon dioxide most of the time? What form is it usually? Right, it's usually in the air. So how are the plants going to be getting the carbon dioxide out of the air? Sort of. It's sort of mesophyll cells. They are specific cells. It's the pores. It's the pores in the leaves. So carbon dioxide is brought into the leaves through these pores called stomata. And so these are pores created by cells called guard cells. So if you were to see this on a leaf, if we have a kind of a larger picture of our leaf here, it would have like kind of like that. So there are two guard cells and they open and close the pore. So it diffuses in through the stomata. And this is a process called gas exchange. So we have gas moving into the leaf, carbon dioxide gas moving into the leaf. And there's another part of gas exchange, which we'll talk about in a second when we get to the products. So the next question, after we know how they get their reactants, would be, where do the products go? So we know that water is also one of our products, right? So where does the water go once we use it, once it gets pulled up through the leaves? So once it gets pulled up through the roots. Because it's both a reactant and a product. So we're using it and then we're also releasing it. So where does it go? Some of it does get stored in the central vacuole, that's correct. It does also get pulled into cells, but over time, the water is actually going to be pulled up entirely through the plant from, let's actually make that blue, from the roots, up through the stems, and actually out of the leaves. So the plant sort of acts like a giant straw constantly. This is the process called transpiration. It's being pulled the water up through the roots, and then it exits through the leaves, through the stomata in the form of water vapor. So water is released through the stomata in the leaves. Okay, so if you actually were to measure the humidity directly next to a plant and then about 10 feet away from the plant, you would see different measurements because plants are constantly going to be pulling water up and letting it out through their leaves. All right, so what are our other products? We have oxygen and our last product, glucose, right? So let's talk about oxygen. Oxygen is the other gaseous component of our reaction. So we know carbon dioxide gets pulled in through the stomata, so our product oxygen, where is that going to be going? Exactly. So oxygen is going to diffuse out of the leaves through the stomata, right? This is the other part of gas exchange. So we are exchanging carbon dioxide for oxygen. Gas exchange occurs in a lot of places. It occurs in our lungs. It occurs through frog skin. It's any place, any time where we are changing out one gas for another gas for a cellular process. And then we have our last product here, our glucose. Now this is arguably one of the most important products or components of this reaction at all. So where is all of this glucose going when we do photosynthesis? You are right about the starch. So it's getting stored and then it is getting used in the citric acid cycle. So glucose, it can have three things happen to it after it gets produced in photosynthesis. So it can be stored in the form of starch or cellulose. It can be stored in a fruit. So it's just stored somewhere in the plant. It can be transported. So we know that plants have this system of tubes to transport the water. They have a similar system of tubes that transports the sugar products. So that's why you get sap from trees. So that sugar gets moved around the plant to wherever it's needed. That's why you can get potatoes in the root systems when photosynthesis is only happening in the leaves, right? So the sugar gets transported. And then the last thing is the other part that you mentioned is that it gets used in cellular respiration. So we talk about how, yes, exactly. So we talk about how the plant has both chloroplasts and mitochondria. So these two processes, photosynthesis and cellular respiration, go hand in hand in plants. So they are collecting that energy from the sunlight, converting it into glucose, and then oftentimes they are immediately using that glucose to fuel the citric acid cycle and produce ATP for cellular processes. So it's this constant feedback loop, and it's really intriguing. Okay, so now we know where each of these things comes from and where they go once the reaction is over. Any questions about how this relates to the equation or how the plant gets each of these things? Any questions? We are currently talking about C3 plants, and I will get into C4 and CAM at the very end. Okay, so now we know what's going on. We know practically where these things are coming from and going. We know all the components of the reaction. So let's talk about how we can measure these things. How can we actually measure photosynthetic activity? So there are typically two ways that we measure activity of a chemical reaction. We can either measure how much of the reactant is used, or we can measure how much product is produced. So when we are looking at our reactants, measuring our reactants, we're typically going to measure carbon dioxide. So how can we measure carbon dioxide? Any ideas? There are typically two ways that we do this. That is a definite answer there. That was the first one I was going to talk about. So you put it in a closed chamber with a CO2 probe. That is one of the best ways to do this. So you place the plant in a sealed chamber. It's going to have everything closed off, so there's no air coming in or out of the chamber aside from what's passing through the plant. And then the CO2 probe detects the amount of CO2 around the plant in that chamber, and it can tell you the change every few seconds. So this is a really good way to detect the change of carbon dioxide over time, and to get a really good quantitative value for how fast photosynthesis is occurring. And the other way that we can detect how much carbon dioxide is being used is by using a pH indicator. And you're going to be doing this in one of your labs. And you're going to be using the plant Elodea for this. So Elodea is an aquatic plant. It lives submerged in water. So instead of pulling its carbon dioxide from the air, it's going to be pulling its carbon dioxide from the water around it. So what we can do is kind of measure the pH of the water around this plant. It's similar to measuring it in the air, but we're using water instead. So when carbon dioxide gets injected into water, it's going to create carbonic acid. So if you've ever had a soda and it's a carbonated soda, we get some acid in that soda, right? That's carbonic acid. So the pH of a solution with carbonic acid is going to be low. It's going to be acidic. And then as the plant submerged in the water starts pulling that carbon dioxide in, it's going to slowly change the solution to be more basic. It's removing the carbon dioxide, removing that carbonic acid, and letting the solution become more basic, right? Okay, so we have figured out how to measure our reactants, our carbon dioxide. So let's talk about measuring our product, the oxygen. So we can measure we can do this in a pretty similar way. So we can use an oxygen probe. So this is the same setup. You put your plant in a sealed chamber with the oxygen probe. It's going to detect how much oxygen is in the air every few seconds. It's going to give you that same quantitative result. And then we can do a similar process using the submerged plant Elodea. And we're going to use a volumeter for this. So this is a tube. I will draw a little diagram of this. So we've got a tube. Inside that tube, we're going to have water, and it's going to be capped. So it's sealed off. There is another tube that extends out of our seal that's got measurements on it. And when we put a plant in here, our Elodea, we're going to see something pretty interesting happen. So this plant is submerged in water, right? So as it's producing oxygen, that oxygen is going to have to displace the water, right? This is a sealed tube. So the water is the air is being produced by the Elodea, and the air is going to collect at the top of the sealed tube. So the water level is going to start pushing down. The air pushes the water level down. Now this tube right here that goes through the seal is the only way for that water to be displaced. So the water is going to be pushed up the tube, and then we can read it right here. So this would be kind of a blown up picture of this. And then we would see the water here. Does that make sense? So we have our submerged plant, it's producing oxygen. The oxygen is moving the water out of the way, and then the water is being measured in the very top part of this apparatus. Okay. We've got just a couple more things. Let's talk about wavelengths of light. So we've talked about how the pigments are different colors, and we know how light is used to provide energy for photosynthesis. But what happens with different wavelengths of light? I know we touched on this earlier when we talked about green chlorophyll. So this is a diagram that you'll often see for this topic. This x-axis is all of the wavelengths of light, and it shows you what colors they are at what wavelength. And then the y-axis is the amount of light energy absorbed at that wavelength. So you can see we've got these two peaks right here for chlorophyll A and B, and then we've got one for carotene, right? And you can see also that there's this massive dip right at green, all the way from green to yellow. So this is from what was mentioned earlier. This diagram doesn't have xanthophyll on it. I didn't see one with xanthophyll, but I'm sure there is one somewhere. Xanthophyll is not one that we talk about a whole lot. Typically, you're just going to talk about the chlorophylls and the carotenoids. It is pretty similar to carotene. So it's going to be the yellowish pigment. It's also going to be picking up the bluish colors. Okay, so we can see here on our diagram, we've got chlorophyll A and B, and they are absorbing a lot of blue and red. So they're absorbing over here in the blue and purple spectrum, and then up here in the red spectrum. They are going to be reflecting green, right? They reflect the green back to our eyes, so we see the plants as green. And then we've got carotene. Carotene is our yellowy orange pigment. And it is going to be absorbing in the blue spectrum here, and it absorbs a little bit in the green spectrum. and it reflects orange and yellow. Okay. So these are how the different wavelengths of light react or are absorbed or reflected by our pigments. So if we were to take a plant and shine a blue light, a red light, or a green light on our plant, which one would you expect to grow the slowest? That's true. The green one might die. So yes, the green one would grow the slowest because the plants can really not absorb green light because of what pigments they're actually using. So it's pretty interesting. I always like that particular chart. It's what's extra interesting, and I use this a lot for my work, is you can actually shine light through pigments and figure out how much pigment is there based on how much light it's absorbing. And you test it at the green wavelength or at the blue wavelength and the red wavelength. It's okay. So let's gotta get to our last topic. So let's talk about what sort of things can impact photosynthesis. What sort of things can impact photosynthesis negatively? What can go wrong? Yep, that's definitely part of it. So if you don't have enough of your reactants, you're not going to be able to fully undergo photosynthesis. Any other ones? Any other guesses? I'm leading up to a really specific one. Okay, so another possibility is not enough light, but there is this actual like intrinsic problem with photosynthesis. And this intrinsic problem is that it involves a lot of enzymes in the mechanic. And these enzymes are supposed to be specific for carbon dioxide. But the enzymes aren't specific enough, and a lot of times they bind oxygen on accident. So if we're trying to grab our carbon dioxide out of the air and the enzymes are grabbing oxygen instead, you can imagine that that would be a problem, right? So this process um, of enzymes grabbing oxygen instead of carbon dioxide is called photorespiration. Um, and it is a huge problem for plants. So much so that two completely different pathways have developed and evolved over time to deal with photorespiration. So, the first pathway that we already know about is the C3 pathway. This is just normal photosynthesis. There's really no fallback here to prevent photorespiration, and C3 plants are susceptible to photorespiration. But we have two other kinds of plants that have adapted and evolved over time to be able to avoid photorespiration. And the first of those is the C4 plant. So these names C3 and C4 have to do with the carbon molecules that get moved around over like during photosynthesis. So the C3 uses a 3 carbon molecule and the C4 uses a 4 carbon molecule. Now, the big picture of C4 plants and how it actually manages to avoid photorespiration is that the C4 plant separates its photosynthesis from its oxygen exchange physically. It separates them by putting them in different places. So you might see a leaf diagram that looks like this. So we've got the edges of our leaves, and we have a tube, and then we have a lot of green cells around the tube, right? So this is a diagram of a C4 leaf. And these are called the bundle sheath cells. So gas exchange is going to be occurring up here, like this, and photosynthesis is going to be occurring here. So they are physically separated from each other. Now this isn't the only way we do prevention of photorespiration. We have another pathway called the CAM pathway. And this name also is associated with a particular carbon molecule, but that's not the easiest way to remember it. CAM, instead of separating spatially, it's going to separate by timing. So it separates its photosynthesis from oxygen exchange temporally, so by timing. So yes, this is where we get like cacti and succulents. So these plants, they are going to time when they change oxygen and carbon dioxide and when they actually do photosynthesis to prevent overlap of those two processes. So they open and close their stomata at different times of the day. Okay, so at night, they open the stomata and they allow gas exchange. And during the day, that should be nighttime. During the day, they close the stomata and photosynthesize. So it's completely separated by the time of day. It also decreases water loss. That's exactly right. So they're very well adapted to really bright, sunny, hot environments like deserts. So those are our two different pathways, the C4 pathway and the CAM pathway, that help prevent photorespiration. Do we have any questions about this? I also forgot to mention earlier that the homework assignment this week, the post lab, is sort of written to go along with what we've just been talking about. So if you want to pull that up, take a look at it and make sure that you understand what's going on in that worksheet. Now would be a great time to do that. You can also double check that you recognize everything from what we've already talked about. So if you have any questions about that, please let me know. If you want to pull up that post lab or the virtual labs and work on them, you are welcome to do that. Otherwise, you are welcome to disconnect, go about your day, and I will see you next week.